Ending's gonna come out in a week. But that is week after.
Yes.
Wait, next week is the critique, and then
Manuscript.
this
Well,
next week.
that's why the things we have to.
Goodness, Micro doesn't have anything like lab experiment. I would do that.
I would do that. I would have literally get 2 hours of sleep.
Yeah.
That would be insane. Oh my gosh, yeah.
It's only supposed to be an hour, right? I can look at it while I set and stuff.
Yeah, probably today is not the day for that.
I did a lot of these last night.
Yeah.
But now I need to take it into gear. Really, it's just
What? We did our game board.
I got the research meeting.
How was it? That's pretty good. Different.
3 hours and haven't done anything
It's
because
just like.
it's everything.
Is it to your class next semester?
That's the before like I did the summer research again, and I'm doing something entirely new. So yeah,
Are you ready?
So how do you guys, how's it going?
something wondering about,
Yeah.
or even triple, because I have capstone and the independent research in my last semester, and then this semester I did. Next upcoming semester I'm doing the physics research, so I'm up to reading.
You talked to us for another 15 minutes.
Yeah, I mean, if I'm doing, but yeah, this is this, this is this, and this is this, and this is this. Or one actual presentation, because I saw so yesterday session I did this like honor conference thing, and then like a presentation award was like this massive glass statue. Like that was really cool. I feel like I want to apply for it, but at the same time, do I actually want it? That's so big, and I have to get back to Japan at some point. I don't want to bring that massive. Yeah. And if these, I heard does made up glass or something, so I don't want to break it because I don't believe in you. So, yeah, or at least, good, looking good on the resume.
Because you do
Not
not
as good
have for
as
it.
the application.
So I don't know. Just like don't waste our time trying to like whatever not to say that.
Thinking about
Making him look worse.
present, past, and future all together.
Yeah. Just like everything.
That's my goal.
Yeah, just one more mistake.
That at the same time,
We might be breaking 100.
but I'm just ready for summer. That's what I think.
Oh, yeah.
Yeah, he just says it, but he
It'll
doesn't
be a nice
actually.
little break.
3 months is enough to read
Yeah, I feel like it just.
the suffocation of 3 months.
Calls yourself down. Did you see the word sports in there? Gabby, bro. Quite simply.
I don't know, maybe.
That was like, yes. You act like we are poking the bear.
I mean, which is fair because.
What are you done with tonight?
Or it's not easy.
Don't wait to the bench. Poke the bear. You talk about his conduct. Like, I would never pull someone off for making mistakes. I would never talk poorly about them. You know, it's like, you literally got in trouble for doing like that. I literally was, what?
Yeah, I was staring at the whole time. It was so funny.
She said, I thought it was still not my life. And she said, would you say that you're poking the bear?
Oh. That's not all.
That was crazy. It was just like,
Yeah, that was.
like that conversation, he just shouldn't have talked after breakfast. He should have just let us sleep.
I'm sure I didn't know. Yeah. Tamika, so you're working now.
You're smarty
Can I see yours? I want to see what I got.
A or D.
Alright guys, I know you're probably now invested in looking at your exams, but let's just put them away for now, and if you guys have any questions about anything, feel free to email me or come up after class and we can talk about it. So, how are you guys doing today? Not bad? Alright, so just a little note. So I was trying to print worksheet 7 for you guys. I've been trying for the past 30 minutes, but the printer isn't working for me for some reason. It's working for other people. I've tried for my personal computer and this computer here, and both of them are not printing. So it's on Blackboard. If you guys are able to, either fill out your answers on it and then print it out and bring it next week, that would be great. And so I'll just see if it ends up printing by the end of the class. I'll have it for you, but if not, then you might just have to print it on your own. Just want to give you a heads up so you don't think that there's nothing due next week on Monday. All right. So today, we're going to be kind of switching gears a little bit, and for the first whole part of the semester, we've been talking about microbes and bacteria, and their mechanisms and how they influence us in good context and in bad context. But there's a whole other piece to the puzzle, and how we interact with microbes, and that is our immune system. So microbes definitely are influencing our immune system, whether it's to keep us in a healthy and homeostatic environment. But when microbes are harming us, then our immune system is the first defense to recognize that something is there. And so, you can't really study one without studying the other. And so today I'm going to give you some background on innate immunity and what happens during infection. This would be some chapters 23 and 25 in your book. But some of these concepts are inherently a little bit more difficult because it taps a little bit into immunology. This is not an immunology course, but just to kind of get an idea of people's levels of knowledge at this point. Has anybody taken an immunology class before? Yeah, maybe one person. Okay, so yeah, as I already assumed, immunology is a whole other topic in itself, and so I hope to give you a good introduction to it in the context of microbes. But please feel free to stop me if anything doesn't make sense or if you need anything clarified. I will say, my work also taps a little bit into immunology, but I'm not a full immunologist, so there's obviously things that I might not know the answer to right away, but I'll be happy to figure it out and get back to you. So. With that said, where's my little thing? My little pointer. Alright, well I can't find my pointer, but that's okay. Alright. Guess we'll still get through it. Alright. So, your immune system is essentially your body's security system, and it starts on the outside. We have physical barrier. We also have chemical barriers. So your skin is, the largest organ entirely on your body. It is considered an organ, and so that's clearly an anatomical physical barrier. Your tears also serve as a barrier. There's chemicals in them that help to make sure that microbes don't get into your eyes. Your stomach acid is also a chemical barrier. I mentioned this many times. The stomach pH is really low, and it serves as a way to prevent bacteria from colonizing. And so your earwax is also a barrier. Mucus, will be a barrier. We talked about it in the context of the gut, how there's mucus layers that protect the epithelium, but also, in your nose and in other mucosal, epithelial barriers as well. Tiny hairs in your nose, those also serve as a barrier. When there's an allergen or something that, gets into your nose, those tiny hairs in your nose really are serving as that barrier, and that's why you end up sneezing and you get that tingly feeling and you have to sneeze. So they're recognizing that something foreign is in there and they need to let it out. So those are part of your body's defense system. But then we have innate defenses, and so innate defenses are innate to you. They are inherent, and they are things that your body's immune system is telling your body that something is going on, and so that begins with inflammation or swelling. There's chemical signals that happen in your body, like cytokines being released and things, and we'll talk a little bit more about that. I'm sure you guys know that if you have an infection, your white blood cell count will be really high, and so your white blood cells are your innate immune cells that will, be able to eat the bacterial invaders in a sense. And so the innate defense is basically this generalized response to all threats. It's not specific, it's just that your body's recognizing that something's going on and you have this influx of immune cells going to the site of infection, and they will respond by causing chemical signals and producing cytokines and antimicrobial peptides and things, and they also will try to themselves, the immune cells will try to engulf and kill that bacterial infection, and then you'll also have inflammation, swelling, fever. Those are all general things that will happen in any infection. So your innate defense is essentially recognizing that there is something wrong and dialing 911 to alert the SWAT team about the invader. So it's more general, and so that is the innate defense. And then there is another line of defense called the adaptive defenses, and so this is more specific. It's a specific response that's tailored to the threat or infection. So this is where your body makes certain cells called T cells and B cells. We'll talk a little bit more about that, and antibodies as well. Within your adaptive defenses, that is essentially is a memory system. So once your body has seen an infection before, then it can have cells that are memory B cells, for instance, and they are always going to be there, and they'll then be able to recognize that specific invader again, because it's basically like muscle memory for your cells as well. So that's why it's more specific. And so your adaptive defenses, is analogous to having the SWAT team actually come and try to, get rid of the invader. Any questions on this? So another, quick little overview of your immune system. The innate immunity, like I said, it's more generalized, and it's more of an immediate response. And so because of this, it evolved really, really long time ago in, invertebrates as well. And so it's very ancient. It's the first line of defense against any foreign pathogen. And so, it consists of anatomic or physical barriers, chemical barriers, but it's non-specific. And this is something that you're present with at birth. Your immune system develops, like we talked about, while the baby or the fetus is still in the mother's womb, and you get some immunity from your mom's immune system. But then your adaptive immunity grows as you grow older, and because it's that second line of defense, it's going to be slower to respond, and so it's more recent. And it's been shown to only exist in vertebrates. So that's telling us evolutionarily that with more complex organisms, you're having a more complex line of defense to the immune system. And the way that it works is it reacts to specific structures called antigens. And so your body has to detect a specific antigen before a response is able to be launched. There's so many different antigenic structures, and this will develop over time after birth. So what's an infection? An infection is when the invasion of an organism's body tissues by disease-causing agents. So basically, some disease-causing agent invades your body, and then that agent multiplies and is metabolically active, and basically an infection is the reaction of the host tissues to the infectious agents and any toxic compounds they produce. So it's the invasion of the organism, that organism being able to multiply or be metabolically active and persist, and then how your body's or the host tissue is reacting to that agent. And so infectious disease is the disease that results from the infection by that transmissible agent. And it becomes a disease, when it's transmissible, when it's something that can be passed from host to host, including via both an abiotic or biotic intermediate. And so what that means is, we know that viruses, for instance, are causes of infections, and they only can survive when they are living on a host, so those need a biotic intermediate, whereas bacteria can also stick on surfaces and things that are abiotic or non-living. And so one thing that's important to kind of distinguish here is that all infectious diseases are infections, but not all infections are infectious diseases. Not all infections end up having, this transmissibility and being able to be passed down, and not all infections give you those disease straits that can then make it transmissible. Does that make sense? So microbes and infection, we've talked about, different microbes being able to cause infection, bacteria being one we talked about mostly, but also viruses. Parasites also are, really, really interesting in the way that they infect us, and parasites can be protozoa, they can be worms or helminth infections. And so there are various modes of entry into the body and methods of infection. You can get something through eating something, scraping your leg and then having that access to that, those epithelial surfaces, and then bacteria can, get in there and cause, a deeper infection. They can be tick-borne infections and transmitted through ticks. So it really depends. And, this is just to kind of give you an idea that infections arise from different, different types of microbes, and then also various modes of entry. And within all infections, they can be described really into two different types, being extracellular or intracellular. So an extracellular infection means that the pathogen lives in the body, but outside of individual cells. That could be in the interstitial space. So what that is basically it lies between your blood vessels and cells. So it's the fluid in the vascular space. So it can kind of just be within those fluid and structural environments that are surrounding those cells, but it's not necessarily in the cells. It can also be in your blood, in your lymphatic system, your lymph nodes, and on epithelial surfaces, but not necessarily inside of individual cells. So here's a good example. You can have right here we see these microbes. This is a virus. These are some different bacteria. They can be, this is the cell here, so they can just kind of be in the space around the cell, or they can be on top of the epithelial surface. And so any organism really can be extracellular and can cause an extracellular infection, like viruses, bacteria, protozoa, fungi, worms. And then intracellular infections, the pathogen will live and reproduce within the cell, so it actually gets into either the cytoplasm, or it can be vesicular and be inside of the cell in its own little vesicles. Questions on this at all? All right. So infection versus disease. An infectious agent entering the body does not guarantee disease. So like we said, an infection is not necessarily an infectious disease. And so organisms have to first breach the anatomic and chemical barriers in order to gain entry. And then it has to survive the innate defenses, and then finally overcome the adaptive immune response in order for it to then be called something that is a disease. It's been able to surpass and get through all of these barriers that our body has put in play for defense, and then you'll get the disease if it's able to overcome all of these responses. So we're going to talk about some passive barriers to infection. This will include the physical barriers, the chemical barriers, anatomical barriers. So kind of just showing you where things would be within the human body. So in your nasopharynx, so here in your nasal cavity, we have these cilia that we've kind of touched on briefly before. They're kind of like these hair-like structures that will remove particles. Skin obviously is a physical barrier. Lysozyme is an enzyme that's in tears, and so basically it dissolves cell walls of bacteria. It's very, well used actually in, the lab to dissolve cell walls and, break open the cell wall of bacteria. So it's actually in our tears as well. And that's, really important evolutionarily because we don't want bacteria to get into our eye. And so that's one way that we are able to, prevent that. And then obviously there's mucus and cilia lining the trachea that leads to the lung. And so, like I said, skin produces antimicrobial, fatty acids and peptides, and there's a normal microbiota that inhibit infection. Your stomach acidity is a pH of about 2. It's really acidic, so it inhibits microbial growth. You have your blood and your lymph that has your lymph nodes that basically have proteins that inhibit microbial growth. Your microbiota, your normal microbiota, your commensal microbiota is essentially exerting this colonization resistance to compete with pathogens in the gut and on the skin. And then once you go from the stomach down to the small intestine, there's going to be a rapid pH change in the small intestine. And so that's going to obviously affect, the number of organisms that can grow in the stomach and then in the small intestine. Yeah, so this transition reduces microbial growth. Your urinary tract, there's bacteria there that are healthy, and that flushing of the urinary tract prevents infection. Epithelial cells throughout the body also have these things called tight junctions that are essentially these proteins that click the epithelial cells together really closely so that a microbe can't necessarily come in and breach those tight junctions and breach the epithelial barrier. So that inhibits pathogen invasion as well. Any questions on those, on that kind of overview? Okay, so within the anatomic barrier, we talked about epithelial cells, and I was talking a little bit about the tight junctions. So that's what it looks like. You have 2 epithelial cells here. This is the lumen, the outside of the epithelial cell, and then here they're kind of stitched together by these tight junctions. And so we've seen that during microbial infection, if a microbe is able to get in through the epithelial cells and make contact with the immune system, which would be down here, you are unable to see. Are you able to see it? Yeah. So your immune system would be down here, and so that would essentially mean when a pathogen invades, it means that it was able to break through this tight junction barrier. And so, there's these linings and tubular structures, not just in the GI tract, but also in the respiratory tract and the urogenital tract as well. In the GI tract, though, that's kind of what we're going to focus on a little bit here. And so there are these gut-associated lymphoid tissues that we call GALT that contribute to keeping things out. And just another, note that this internal epithelium is called a mucosal epithelium because there's a big mucus layer on top, and so these are specialized because they secrete mucus. So when I talk about the internal epithelium, always remember there's a mucosal layer, and that's also termed the mucosal epithelium. So how's the body protected from invasion by microbes in the gut? We have this system called the gut-associated lymphoid tissue. And so basically this is a component of the mucosa-associated lymphoid tissues, which works with the immune system to protect the body from invasion in the gut. So here I'm showing you a Peyer's patch. It's just a different, I guess, slice of how we're looking at it. And so over here in the intestinal cells, you can have these Peyer's patches that are just part of the epithelium. And so it's there in those Peyer's patches that kind of guide the immune cells below it. So here I'm really struggling to show you guys what I'm trying to show you. Let's see. me try to do it this way. Okay, so yeah, there's these Peyer's patches, and essentially once, you know, the bacteria are in the Peyer's patches, then there are immune cells that are going to be like on the outside, and it's the Peyer's patches that kind of connect to the immune system and tell the immune system that there's something here. It's kind of that bridge. And so here what I'm showing you is there are antigens on the bacteria, or just the bacteria. They're taken up by these M cells. Basically, these M cells are a part of the Peyer's patches, and so they basically are taken up. So over here in red, you're seeing the bacteria, and so they're taken up through these cells here, and then on the other side there are these macrophages, or these immune cells. So the M cells are feeding the bacteria to the macrophages, and then that will present the antigen to your other cells. The macrophages will take this antigen or bacteria to other immune cells to create an adaptive immune response. But it's through this M cell within the Peyer's patches that facilitates this crosstalk to your immune system in order to tell it, you know, that there's something here and I'm presenting it to you. So just to give you a little bit more definition, Peyer's patches are a group of well-organized cells in the lamina propria and the submucosa of the intestine. And so this M cells are specialized intestinal epithelial cells found in the Peyer's patch within that gall tissue. And so lymphocytes here are white blood cells, and then over here I'm showing you macrophages as well. And so those basically are phagocytic, so you can see that they engulf the pathogen over here. They're really just kind of like trapping it and engulfing it, and then they can present it to other cells. Any questions on this? All right, so enzymes, basically, you know, these are your tears, your saliva. Lysozyme is the one we talked about. It occurs in tears and in certain body tissues, and it basically destroys bacteria by hydrolyzing the polysaccharides in the cell walls. And hydrolyzing, just a reminder from Chem 101, means to break down by chemical reactions with water. So it's breaking down the polysaccharides in the cell walls, and that's how it destroys bacteria. Phospholipase is another enzyme that is a chemical barrier that also exists within our cells, and that also hydrolyzes phospholipids of bacteria, hence phospholipase. Acids in the stomach, there's digestive enzymes, bile salts, and fatty acids that are able to kill bacteria, as well as antimicrobial peptides that are produced by our immune cells, and those consist of defensins, cathelicidins, and histatins. And so you don't really need to know the various like ins and outs of what each one of these are, but they are antimicrobial peptides. And so I'm, kind of going to give you a lot of terms. I've shown you, already Peyer's patches, M cells, and epithelial cells, mucosa. So I really want you guys to, keep things straight. Some of them are interchangeably used. And then within the intestinal epithelium, there are also these Paneth cells. So they are usually in the crypt, so in this kind of valley area in the cell, and they contain an abundant amount of antimicrobial peptides here. So that's just kind of giving you a little bit more context as to the types of cells that exist in our epithelium, and they have specialized structures. Some of them, like Paneth cells, will have a lot of antimicrobial peptides. There's others called goblet cells that produce mucus, and so they're just specialized types of epithelial cells. And so what you do need to know is that antimicrobial peptides, or AMPs, are small peptides with potent antimicrobial activities, hence the name. They're produced by many cells in the body, including neutrophils, which we'll talk about, type of white blood cell, and cells of the mucosa, like the Paneth cells. And then many antimicrobial peptides will display a direct and rapid antimicrobial activity by just disrupting the physical integrity of the microbe's membrane, or it can translocate across the membrane into the cytoplasm of the bacteria to act on intracellular targets. So it can, just disrupt the outside of the bacteria, or it could actually go through the bacteria into its cell membrane and act on its intracellular targets. And like I said, there's categories of natural antimicrobial peptides, there's defensins, and this is just kind of to show you that there's a bunch, and they are produced by different types of cells in your body. And so other species also, will form these antimicrobial peptides. So it's just showing that this is an evolutionarily conserved part of the innate immune system. Any questions at this point? Okay. So antimicrobial peptides and the intestinal epithelial barrier. I clearly love to talk about this. So this is kind of just showing you another, image of what the small and the large intestine looks like. We've talked about differences in these structures. The small intestine has just one layer of mucus that you see here in yellow, and within this mucus layer you have these antimicrobial peptides and things that are helping defend against foreign pathogens. But then in the colon, or the large intestine, there's two mucus layers, and there's these microbes that are commensal, that they're really abundant here versus what looks like the amount is in the small intestine. And so, there's different types of cells. There's Paneth cells, goblet cells. Like I said, they produce mucus. Enterocytes, stromal cells. There's stem cells in this area as well to regenerate epithelial cells when they are destroyed or they are disturbed. If you see IEC, that just stands for intestinal epithelial cell. So as a quick summary of these passive barriers to infection, we talked about all of these things. I'm not going to keep repeating them, but these are a combination of physical, chemical, and anatomical barriers. So that's, kind of some of the, main passive barriers to infection. And so we're going to kind of talk a little bit more now about the immune system alone. So where can we find immune cells? Anybody guess? Everywhere. How do I study your immune cells? If I want to know if you have an infection, what do I do? What does a doctor do, not me? Physician. Yeah.
Really responsive, like information.
Yeah, absolutely. Definitely. And then if I want to know what kind of infection you have, what do I collect from you? Swab is one. Sorry? Blood. Blood. Lots of immune cells in your blood. So you withdraw your blood and you place it in a tube. And then once you do that, you'll centrifuge it, which means you spin it down really quickly to kind of separate the different layers and densities within the blood. And so within your blood, there's also plasma. So I'm sure you guys have heard of not just blood, donations, but you can do plasma donations, because your plasma contains about 55% of your entire blood. It's not dense at all, and it has a lot of, really important antimicrobials and immune cells in there. Then there's this buffy coat right in the middle that separates the two. And then your actual, the red part of your blood is only less than 1% of the entire blood. And so that really is, that's the buffy coat. The buffy coat's less than 1% of the whole blood. But then the erythrocytes, which are the remainder of your immune cells, that's the most dense component, and that's 45% of the entire blood that you have. So here's just a picture of blood that's not stained. Within your blood, you have white blood cells and red blood cells. So here I'm showing you the white blood cells with just the arrowheads. They're generally larger than red blood cells. They're colorless, especially because they're not stained, and they're more granular than the red blood cells, which are here kind of look a little bit redder, and that's because they are, red tinged from hemoglobin. And so yeah, you can see the differences in the sizes as well, but your blood contains both white blood cells and red blood cells. And so leukocytes are your white blood cells. So this is a stain that was done, and so leukocytes are just many different types of white blood cells. So there's neutrophils, which we also call PMNs, or polymorphonuclear neutrophils. There's eosinophils, there's monocytes, there's lymphocytes. Lymphocytes are your B and T cells, and we'll go through these, but just to give you a little bit of, an introduction here. Neutrophils are the most abundant innate immune cell that you can have. Eosinophils, you can see here, they contain these granules, and so these are readily stained, so that's why you're able to see them really well. That's one way when we're doing histology, we're able to stain certain components of different cells to be able to identify what they are. Monocytes are just like a simple oval, nucleus and cytoplasm. And lymphocytes, they occur especially in the lymphatic system, hence the name, and so those are your B and T cells, and we'll go through those a little bit more. Just a picture kind of showing you there's so many veins and arteries in your body, and so the circulatory system, contains of the arteries, veins, and capillaries, and so this is, where your blood is occurring and where you have all of these different immune cells. But this blood cell production process has to start somewhere. It didn't just exist in your entire body. So how does your body, make certain blood cells and know where to take those blood cells? There's a whole system that occurs, from really birth and even before then, and that's called hematopoiesis. It's the blood cell production process. So all cellular blood within your body, every component of your cellular blood is derived from the hematopoietic stem cells. And that all starts in your bone marrow. And so we're kind of gonna go through these. Might kind of be a little intimidating to see so much all at once, but I'm gonna try to go through this as slow as possible. So yeah, in your bone marrow, that's where you have your stem cells, and so that's where, they're pluripotent, which just means that they can differentiate into different types of things. And then within that, these stem cells will then branch out into either a lymphoid progenitor or a myeloid progenitor. And so basically what that means is that these are just two different subtypes of these hematopoietic stem cells that will then give rise to more specialized and different types of blood cells. So your myeloid progenitors really end up giving rise to your red blood cells, granulocytes or neutrophils, your monocytes, platelets. I'll talk about that a little bit more. And the common lymphoid progenitor really gives rise to your lymphocytes, your B cells, your T cells, NK cells, which are natural killer cells, ILCs or innate lymphoid cells. And so then those will then differentiate into effector cells, which actively respond to stimuli and affect some type of change. So that's where the name effector comes from. And so this is really showing you the adaptive immune response, because you have these B cells and T cells that are then being, differentiated into even more specific cells that have to be activated. And so that's really where they're responding to the innate immune system, saying like, here's an antigen, we're presenting it to you, like you need to now, the innate immune system's calling 911, and this is the SWAT team that's coming to really get rid of the infection. And so your myeloid progenitors, though, will give rise to more so innate immune cells, so those first responders. And so that's where you'll have neutrophils, eosinophils, basophils, monocytes, and then those can then be differentiated into cells that are going to be there residing within the tissues that are going to be that first point of contact with the microbe. And so those are your mast cells, your macrophages, your dendritic cells, and so we'll talk a little bit more about those. But any questions at this point? Okay. So just another way to show you this, this is the immune cell family tree. So everything starts from the hematopoietic precursor stem cells in your bone marrow. And then you have things kind of branching off into either a myeloid stem cell or a lymphoid stem cell. This is still in your bone marrow. And then, your myeloid cells can then differentiate into other things. We call these polymorphonuclear leukocytes, or PMNs. Or PML in this case. And then, you have just all of these different types of differentiations. You don't have to remember these exact steps, but you should know, the types of cells that will be derived from myeloid stem cells. And then from the lymphoid system, you can have both innate immune cells that can arise, like natural killer cells, and I mentioned on the previous slide, your ILCs, and innate lymphoid cells, but then you can also have them differentiated even further into your T and B cells, which are part of the adaptive immune response. And so that is when things will then either be processed in the bone marrow or they will be processed in the thymus. So your T cells are processed in your thymus, which is in your thyroid glands. And so your B cells stay and are processed in your bone marrow. So that's where things kind of get moved into your body and can then be differentiated in different places. So like I said, leukocytes are white blood cells, and they're formed by differentiation of the hematopoietic stem cells in the bone marrow, and there's these two main lineages. The myeloid lineage leads to the innate immune response, or innate cells, and lymphoid lineage also results in adaptive response, but that's also, it also can lead to natural killer cells, which are part of innate immunity as well, but generally lymphoid lineage will lead to the adaptive immune response, which are the B and T cells. So we mentioned these polymorphonuclear leukocytes or neutrophils, and so PMNs or granulocytes, so that's neutrophils, eosinophils, basophils, there's monocytes, macrophages, dendritic cells. So a dendrite is basically something that bridges two different things. So a dendritic cell will bridge two immune responses. And so that'll make sense a little bit when I tell you that they are antigen presenting cells. Mast cells, and then we'll talk about natural killer cells, which, like I mentioned, they are part of the lymphoid lineage, but they are part of innate immunity, so that's the exception to the rule there. We'll kind of go through each of these. But first I want to talk about phagocytosis. Phagocytosis is the process of an immune cell engulfing a microbe, and so the way that it does this is, if you look at number one, bacteria will bind to the surface of the phagocytic cell over here, and then this phagocytic immune cell will make these extrusions, and so we call these pseudopods, and so basically they'll extend themselves and try to engulf this pathogen that's adhered, and then once it engulfs it and it ingests it, this basically will create an invagination of the membrane, and it'll form this phagosome that just will contain the bacteria. There are certain enzymes that are produced, like lysozyme, that will want to break down this bacterial membrane. And so once it's engulfed in the phagosome, then it can be killed by lysozymes. And so then the lysozyme can then fuse and deposit, those enzymes into the phagosome, and then, destroy the organism through generating these reactive oxygen species. And so that's just the way that it destroys the microbe, and then it releases microbial debris. And there are certain cells that have phagocytic capabilities. Those include macrophages, monocytes, neutrophils, and dendritic cells. And phagocytosis, though, is primarily carried out by macrophages and neutrophils, and so we'll talk about this. Let me see if this will work, because lately, yeah, it might work actually. Here I'm showing you attachment of a phagocyte to a microbe, and that could be mediated by a specific receptor interaction or charge interaction between the two. So this is the phagocyte here. This is the bacteria, and you can see how it literally engulfs it. This is, an actual image in real time showing this process. It's really good at finding, what it's looking for and then engulfing it. And the way that it does this is because the bacteria probably has something on it, and that this macrophage can essentially recognize, and this macrophage has these receptors that are able to recognize that thing, which we call an antigen on the bacteria. So just to play that one more time. Okay. So phagocytosis, killing of ingested microbes. So just to give you, I gave you the general, very brief, image showcasing this, but essentially, like I said, the phagocytic cells will use these reactive oxygen radicals or species that will kill the ingested bacterial cells because they basically oxidize key cellular components of the ingested microbe. And when this this burst happens, it's a series of catalyzed reactions in this phagolysosome, because remember, it makes this lysosome inside of the phagocytic thing, so we call it a phagolysosome. And so, there's different types of reactive oxygen species like peroxides, nitric oxides, hydroxyl superoxide that will occur. And it's important to know that this occurs within that vacuole in the phagocyte, but that phagocyte, so that cell, is not damaged by those toxic oxygen products. And so those degradative enzymes are only found in that phagolysosome, that thing that has trapped the bacteria, so that the cell does not get destroyed itself. So this is really a way that macrophages kill ingested microbes, but through this phagocytosis. But neutrophils also do this, but in a little bit of a different way, and I think that this is actually really cool. This is something that has been really shown recently in the past few years. But neutrophils don't necessarily act like macrophages where they chase and then they engulf something and they form this phagolysosome. Neutrophils throw these nets, and literally the name is probably made for it to fit the name net, because they're throwing these neutrophil extracellular traps. It's basically, here we're seeing Klebsiella, this is a bacteria, and so outside here there's obviously this neutrophil, and so these neutrophils will throw these lattices, they'll spew a lattice work of DNA that contains antimicrobial compounds into the immediate area, and so it literally looks like a net. So they made it termed something that will fit that. So basically, that net will trap the pathogen and prevent it from spreading, and those antimicrobial peptides that are thrown out into that area will kill the microbe. It's a form of cell death, and so there's certain organisms that can have actually evolved to escape this, and so they then secrete extracellular nucleases, or things that can then break down DNA of the antimicrobial compounds from the neutrophils. So it's important to know that there's a distinguishing factor in killing microbes there. Some other leukocytes are basophils and eosinophils. So basophils play a role in inflammation and in wound healing. And so these basically will release products that are toxic to the microbe, and they'll release chemical mediators that affect the permeability of blood vessels. So what's important though is that we know that basophils and eosinophils actually play major roles in allergic responses. And so basophils have a role also in inflammation and wound healing. When you get a scrape on your knee and it's starting to scab, there's a lot of basophils that play a role in that. Inflammation can also be good. It's not necessarily bad, because then that will trigger wound healing. And so eosinophils play a role in a lot of parasitic infections like helminths or worms, and they also play a role in allergic responses. So they have multiple roles. Mast cells are similar to basophils in structure. So here, if you see, basophils have these granules inside, and so do mast cells. So they're similar in structure. And so mast cells also play a role in inflammation and in wound healing. They're associated with allergic responses because these granules contain histamine. And so once a mast cell recognizes something that is, you're allergic to, it's going to release these granules into that environment that contain histamine. And so that's why when you have allergies, you take an antihistamine, because you're basically trying to, prevent that histamine production that's causing that allergic reaction. Does that make sense? Any questions so far? So monocytes and macrophages. So we talked about macrophages engulfing bacteria. They're phagocytic. They're widely distributed throughout the body. And so macrophages are likely some of the first to contact pathogens. They are part of the innate immune response again. And so, they phagocytose microbes and other foreign particles to clear them. But sometimes if a macrophage, sees that there's something going on and it engulfs that, bacteria or the antigen on the bacteria, and it doesn't really know what to do with it, it can present it to cells of the adaptive immune response. And so macrophages are known to be antigen-presenting cells, and so basically they present those antigens from the engulfed and degraded microbes to the adaptive immune cells. So it's an APC, or antigen-presenting cell. So it serves as a bridge between innate and adaptive immunity, essentially. And so dendritic cells, I also kind of foreshadowed to this. It really is also, a connection between two things. That's why you have these dendrites. These are also antigen-presenting cells. These are present in your skin, your mucosal tissue, your lymph nodes, and your spleen. They can also phagocytose microbes by taking up small antigens from their surroundings, but they have more of a mechanism of just presenting it directly to the other immune cells of the adaptive immune system, rather than acting like a macrophage and just engulfing it and killing it through its own mechanism. Doesn't mean they can't, but they are also really good at presenting these microbes to other cells. Natural killer cells are different from antigen-presenting cells because they just kill, and so what they are killing, though, is they're killing the host cell instead of the microbe. So this is a version that, or these cells actually are derived from, we talked about the lymphoid progenitor system, and so natural killer cells really are able to target intracellular parasites and cancer cells, and so they are able to recognize that there's something wrong with the actual cell, the host cell, and then, or if the host cell has a parasite inside of it and it can't get to it, then it'll just kill the actual cell itself. So they are able to recognize changes in cell surface proteins on your actual, on your immune cells. So if they recognize that something is different on your immune cell, and one of those things is the major histocompatibility complex, or MHC1. We're not going to go through that today, but they basically will recognize changes in the cell surface proteins. And so what they do is they degranulate, or they release their granules onto that target cell and kill it, triggering apoptosis or just automatic cell death. Yeah, so it's antibody dependent, but I'm not going to go into that. So that's kind of an overview of just like the different cell types, what progenitors they come from, and some of the differences that they do, and how they, protect our immune system and our body from foreign invaders. But let's talk a little bit about inflammation. So inflammation is a critical innate immune defense from microbes. It provides the opportunity for these phagocytic cells to leave the bloodstream and patrol infected tissues. So inflammation is involved in extravasation or diapedesis. What that means is essentially what is written right before that is this leakage or movement of the leukocytes or white blood cells out of the circulatory system in the blood and towards that site of tissue damage or infection. And so once those cells are kind of going to the site of infection, from the blood, that's why it gets really red, because those cells are coming from the bloodstream and they're like, oh, there's something going on over here, so let's go over there and try to, you get this vasodilation and you get this redness and swelling. And so there are 5 signs that, are known to inflammation. Heat, obviously, when you touch it, it'll be hotter. Edema, which means swelling. Redness, pain, and just altered function. The cells just look very different. So your inflammation process, here you have your epithelial cell, and so you have a splinter that goes through, and the splinter has these microbes on it, so you get an infection via the splinter. These are pathogens. Over here you have your phagocyte, which is a macrophage in this case. So these resident macrophages, this one's in the bloodstream, you can see. And so this is the blood vessel, or the capillary artery, and these macrophages are then able to, get out of there and engulf the pathogen, and they also will release chemicals like cytokines and alarm signals. So they'll engulf the pathogen here. You can see it's extravasating, so basically it's leaving the bloodstream and going towards the site of infection. So these vasoactive factors and cytokines help deliver even more phagocytes than cells can communicate. So when one is going there and it's releasing these cytokines, those cytokines are a signal for other macrophages and other immune cells to also come to the site of infection to then also produce their own cytokines. So they kind of call their buddies over, and they're all going there to that site of infection. And then some cytokines already, have functions in aiding in that healing process as pathogens are destroyed. And so that's how your, body reacts when you get an infection from a splinter, for example, and in the healing process. So this would be the scab that's going on. Fever. Obviously, fever is, heat in the human body that's produced as a consequence of, certain infections. The normal body temperature range is about 97 to 100 degrees Fahrenheit. 100 degrees is clearly a fever, but it really depends on the individual. And so, like, for people that don't have a spleen, for instance, and I give this example a lot because I don't have a spleen, and so I, know a lot about some of the functions of that. Like for me, if I get a fever that reaches 99 degrees, that's like equivalent to 103 for somebody that has a spleen. So it just depends person to person. But essentially, if you have something, for a normal immunocompetent person, 100.4 degrees Fahrenheit is technically a fever and above. And your hypothalamus in your brain controls your temperature. Pyrogens are a group of just chemically diverse substances that will cause your fever and shock. And so the reason why you get a fever is because that elevated temperature helps to reduce microbial growth. A lot of microbes have an optimal growth temperature at 37 degrees Celsius, which is essentially the body's normal temperature. And so those elevated temperatures help to kind of kill the bacteria or reduce microbial growth. It reduces microbes from growing at all. At least the ones that optimally grow at 37°C. All right, so we'll get through a few more slides here. So microbes have unique structures on them that are immediately tagged by the innate immune cells or immune system as foreign. So your innate immune cells have these receptors that I mentioned that will basically be able to tag and trap the microbe. Those receptors are called pattern recognition receptors. So your immune cells are trained to recognize patterns of receptors on microbes. And the microbes have a unique structure called microbe-associated molecular patterns or pathogen-associated molecular patterns. We call these either MAMPs or PAMPs. For the rest of the talk and for the rest of the class, I'll call them MAMPs, but those are used interchangeably. And so MAMPs, these microbe-associated molecular patterns, they're on the microbes, they're recognized by these pattern recognition receptors that are located on or within the host immune cell. So those can be toll-like receptors. There's a lot of TLRs on and within the cells. There are nod-like receptors, so those are just the receptor types on your immune cells that recognize MAMPs. And so to give you a little view of how MAMPs are recognized by immune cells, so over here we have this whole thing as the immune cell, and so over here we have these toll-like receptors, so these are the pattern recognition receptors or PRRs on the immune cells, and they recognize specific structures. So here they can recognize the flagella from a bacteria or LPS from a bacteria or lipopeptide or lipoteichoic acid. We talked about that being part of the bacterial cell wall. And so these are MAMPs, these are the TLRs or the PRRs. And so this signal, so they basically will bind and a signal is sent to the nucleus over here to induce cytokine synthesis. So then some cytokines are going to be produced to kind of signal like, oh, there's some sort of, recognition of, a pathogen. And so then, then these are nod-like receptors, so just other PRRs. So these are extracellular, these are intracellular. These will bind the intracellular MAMPs. So here, if we have an intracellular pathogen, it's going to get into your cell. And so there are these nod-like receptors inside of the cell that will recognize these MAMPs. And so then these are sometimes activated to make more cytokines, which is just what they do. And so these then can then be taken to, or can trigger this assembly of something we call an inflammasome, which is associated with either more cytokine production and inflammation. So basically it's just how cells talk to each other to say like there's something going on, keep releasing these cytokines or chemical signals to kill the infection, the bacteria causing the infection. And so that's kind of what's going on here. But just to show you that you can have an extracellular structure on the pathogen that's recognized by these toll-like receptors, or you can have a pathogen that gets into the cell that is then recognized by nod-like receptors. These are all PRRs, or pathogen recognition receptors. And they both can then activate, the synthesis and release of cytokines and through just what they do. Does that make sense? So cytokines are messages that tell cells what to do. They're proteins. They bind to a specific receptor in theory, and they signal intracellular, molecule communication. They can act locally or systemically. They can have multiple effects depending on the target cell binding the cytokine. So basically, if there's a threat detected near the breach, seems sus, we're going to call some neutrophils, and then, thanks, and then the neutrophils are on the way, and we know that because they're basically releasing some cytokines. So cytokines are produced by many different types of immune cells. So, there are stimuli that can produce cytokines, and then those cytokines can then be a receptor that can then bind another target cell that can have another effect. So production of cytokines is tightly regulated, and like I said, they can have local and systemic effects. And so they turn immune responses up or down. So they can be produced by cells, but they can also act as receptor signals. So here's a receptor on another cell. They can, they can fit into the receptor and signal for an effect as well. And so just like anything else I've said, too much of a good thing can be bad. out of control cytokine production can be lethal. We call this a cytokine storm, and so that's when you have hyperactive immune cell activity. This can happen with autoimmune disorders, and you have leakage of your blood vessels, you have multiple organ failure and death. This is actually one of the major causes of death from COVID. It's when you have this cytokine storm, your body just does not know how to control it and handle it, and so you get so much production of cytokines, and that can actually cause organ failure and blood leakage, and so that can, lead to bad things. So regulation and is really, really important in this case. All right, that's all I have. Please make sure to print out worksheet 7 and bring that to class next week.